We'll start where we left off, kind of. I'll just follow the PowerPoint, and then I kind of circle around and make sure I get everything else in that I want to get in. So let me get over there to where the PowerPoint is. Yeah, I'm doing that. Yeah, the recording's going. I forgot the other day with one of my classes, which is just terrible. So I know we did this slide. Okay, to me, the only reason I'm going over it is to remind you to become familiar with using the terms eukaryotes or eukarya would be a sort of a generic term, or prokaryotes or prokarya, because I use them all the time. And I'm going to go over, I will go over in detail before we end this chapter, really to highlight the distinguishing features between eukarya, bacteria, and archaea, and go into the different subgroups of eukarya, like fungi and yeast, which are important, protozoans, algae, things like that. So I'll give you the highlight elements of what makes each of those important. So that just from a review perspective, because I know that's material that's on inevitably on the first exam. I know all the starting material on the first exam is like where we end. It's a question of what completes it. And again, just don't even worry about a slide like this. It just gives you an idea of how the domain theory works. And the whole idea when I did that number with 70, and those are important terms, 70S and 80S, because it's one of the definitions. And you'll see that again when we go into the actual anatomy chapters, which are in this textbook 3 and 4, where you really talk about, the distinctions between them. So 70S, S just means it's a fancy name. It's called the Svedberg unit. I just use the term S. There's no need to remember another proper name, not that important. I'll write it down anyway. So again, those are the so-called S units. And so in the prokarya, 70S. And in us, or cells like us, 80S for eukarya. The big deal, and we will get to it, is a really important part that comes in a subsequent, shortly in the next chapter. It's called endosymbiotic theory. And as they will point out as we're going over today, when you use the term theory in a science classroom, it's not like I have an idea. Theory is something that has been tested. It's like the theory of evolution or cell theory. It's been tested. It's almost at the stage of becoming a law, like Newton's laws of gravity. So a theory is a big thing. It's not like I have a hypothesis I'm going to go test it. It's been tested. So it becomes somewhat of a nomenclature is important in that regard. Nevertheless, this endosymbiotic theory is going to kind of show you how our cells kind of evolved from absorbing lesser cells. It's like prokaryote cells absorbed one another and incorporated each other and magically became, and that's sort of where membrane-bound organelles. And a lot of that, and the key piece to that, there are two, but maybe the key piece is that 70S ribosome. And again, those are about ribosomes. The key part about 70S ribosomes is we got them in our cells. And that's the point I tried to make before, why particularly classes of antibiotics that we get to down the road, and this gives you the idea of how it works, is why antibiotics that target protein synthesis, target ribosomes, have a significant degree of side effects. Of this, I have a lot of information. That goes on to the clinical side of things, and you'll learn a lot about that, because if you're going into a healthcare field, oh boy, medicine, side effects, big issue. Nevertheless, so bacteria and archaea are not predominantly, they're single cell for our purposes. The whole difference between archaea and bacteria, really there's two. Big plan. This is all important stuff I have reiterated, even though I'll go over it several times. The big difference is bacteria live everywhere. It's unique, and it's the most important word I can tell you. Big star, underline, highlight, whatever you do. Well, it's unique. peptidoglycan, that chemical that makes up the cell walls, all of them, not an equal amount. They have peptidoglycan. They live everywhere. That's the big deal. The other thing, when you come to archaea, these that live in extreme areas, extreme environments. This bad weather that's coming our way is nothing. It's not archaea weather. Oh, inside a volcano on the oceans. So extreme environments, and they do not, but they have something, they have what's called pseudo peptidoglycan. It's different. Not the same. Looks like it. And so that's really about as much as we're going to cover with archaea to understand the distinction between archaea or archaea, however you want to pronounce it, and I've heard both, between that and bacteria. Our focus will be bacteria. You just want to get that in. You carry it to our cells. You just spent the last term studying our cells, and you're still studying it, I'm sure. Okay, the ones that typically they're going to be either single or multicellular. I'm going to get to the classifications, but probably not today. They can develop into highly complex multicellular organisms. Us, we're not. Okay, I don't think my dog is that complex, but he's got a lot of cells. My wife says I should bring him in one day. I'm toying with the idea. My schedule's a little more friendly. It's a very large dog. It's a very nice dog.
Just show us the pictures.
Yeah, you've seen the pictures.
But we want more.
Yeah, I'll work on
it. All of them.
75, 80 pounds of dog. So, I mean, I'm picking him up at Camp Bauer because we were away up in Cranberry. And this lady goes, "Oh, is that a giant poodle?" I said, "No, it's an Irish doodle, Irish setter and poodle." She goes, "He's gorgeous." And so I'm walking away, I'm going, "Take me an offer." And she goes, "What?" And I said, "Just joking." Okay. It's like you go to a garage sale and you go, "Boy, it's a nice garage. Make me an offer." It's a garage sale. Larger size, small minority compared to bacteria and archaea. Okay, I'll buy that. Enough. And here, okay, I'm not getting into the evolutionary stuff about this. I'm in a weird mood. I've had an odd morning this morning. Okay, and so fine. Okay, insects came before us. They're still here. Reptiles, man, who cares? None of that. Not asking any questions about that. When I do the lab, and I've done that lab 1,000 times, literally, hundreds of times over the years, my first lecture is about what's called ubiquity. Ubiquitous is a great word. It means it's everywhere. Okay, so when something is ubiquitous, and that's I told you, they're all over your skin, they're all over the room, they're all over your shoes, they're all over the carpet, they're everywhere. Every single place. I understand you can't avoid them. I mean, you can every 5 seconds go over and hit the Purell, hit the hand sanitizer. All that's mostly what that's going to do is to denude or get rid of the natural oils that are protecting your skin. I'm going to say this a lot to you folks as time goes on. Most of you are going into fields where you're going to have to do hand care all the time. Your skin is your protection. This is part of this course. If you're going to be working around sick people, you're going to be encountering diseases. You have to protect yourself as well as protect others. Okay? Because you can transmit it. Your hands are the big player. So you have to do everything with regard to your hands. I tell all the students the same thing. Before we were surgical residents. Surgical residents lived for the idea of getting in the operating room and doing surgery. But before we could go in there, every morning when we were on surgical rotation, the attending, these were old school attendings, okay, said, "Okay, show me your hands." And you had to sit there like this. If you had a cut or a nick or anything on your hand, you weren't operating that day. You were a runner, you were going to the floor, you're going to the emergency room, you're doing all that type of stuff, answering the phone. You wouldn't have the fun of sitting in there and cutting on something. Because you became that increases the risk, even if you're gloved and scrubbed, to patients potentially, not just considering yourself. So the hands are so important, and that's what I mean. And we're crazy for this stuff. I used to do this routine. But you like, and nowadays, and worse because of COVID, you see, it's very accepted. You go up there, you're walking down the hall, you're right? Somebody to get along with, but that's why it's obsessive. But you have use lotions, use moisturizers, and not all creams are moisturizing creams. You have to look at ingredients. There's a lot of good products out today that you can use. Okay? Be very, very, really, because you're going to end up washing them a lot. My ex, my first wife, washed her hands so incessantly. If you didn't know, most of you know, she's an RN with a BSN. She had nurse in an emergency room back in the day when we met a long time ago. And she was doing some part-time nursing when our kids were little, and she was doing OBGYN and doing Lamaze instruction. What happened was, I mean, her hands broke down because of this winter cold weather and the nature of her skin, to the point where she could only wash her hands 3 times a day. She had to keep them in gloves and wash the gloves. And we had 3 kids in diapers. That's how bad her hands got. It was every winter. It got progressively. As you get older, it gets particularly worse, particularly for people who are more fair complected, like some of you. And her skin was not quite as fair as some of you are. Crazy. So I know I get off on a tangent, but this is kind of the stuff. I don't have a set agenda whenever I do this class. I go with the flow, and I want to get as much of this in, and we'll get it all covered. That's what I always do. But I hope it keeps it interesting. And right now you're showing up. Is that because it's early? Oh, come on. I know. I will get the Friday Club. You know who you were. No laughing. It's all right. It's not my first rodeo. I've been teaching for 25 years. I've seen this. 2 careers. Okay, ubiquitous everywhere. Crust, polarized scabs, bodies of plate, everywhere. We are filled with them. Your entire alimentary canal, effectively less in the stomach, but your large intestine is teeming. There are probably 10 times more microorganisms, single-celled microorganisms in your gut than you have cells. It's an immense number. You'll see these numbers, they'll come at you. All of this, and they are essential to life. And that's part of what we'll get to with the things and the charts and stuff like that. The accumulation, this is evolution, changes, descent with modification. I'm not going to ask anything about that. But again, it's interesting. Historically, Darwin had the right idea. His examples in the Galapagos off of South America probably weren't the best example of it. There's a lot of things that we see in. You might remember when we'll go over some of these things. You might remember that thing. I know I didn't make a big deal of it, called the 9+2 thing for flagella and cilia. Maybe that's ringing a bell for you. We see that's part of it. These little things in pond water. Their flagella have the same thing that our flagella and cilia have. That's one of the things. Their DNA is like our DNA. Those are the kinds of things that are more about evolution, descent with modification. Okay, that's as much. I'm not going to ask any questions about evolution. They're there. And you can see it. So theory, the whole idea, years and years of testing, nobody cares. Not just a random guess. It's all about this. The whole idea, remember, basic tenet of science, well, the energy on our planet is just here. Maybe we're generating more energy from the sun by way of photosynthesis. And by the way, that's mostly microorganisms. And again, this is the best example of how important they are. All 70%, 75% of our oxygen on this planet comes from algae. Algae are a unique microorganism. Algae are sort of like a microscopic plant. They can be single or multicellular. And the unique thing, again, that key word of karyotic microbes, they can be single or multi-cell. And they have chlorophyll. That's the big player. That's the unique thing. So they do photosynthesis. it probably came originally, you'll see, from photosynthetic bacteria when we get to that endosymbiotic theory thing. But the planets, they're in water. They're water-based. So unique about that, not unique, what's interesting about them rather, water-based, they have a cell wall. So they're kind of like microscopic plants. You want to think of it that way. And they can get large. So algae can be visible when they become multicellular. But the vast majority of this textbook says 70% of our oxygen on the earth comes from organisms. So the reason you're breathing, again, ours probably is coming from the green stuff we have out here. Can you read it? It's okay. I mean, you can take pictures. I have this stuff. I mean, I'll probably have a handout with this stuff someplace. Okay, if I'll find something. You know my handwriting. So so the microbes. This is energy. So think what happens? We see it all the time around here with all the deer populations. We see roadkill, and you'll see the roadkill go through. You'll see rigor mortis. Many of you have seen that when it's like that on the road, and all of a sudden it begins to deteriorate, and then it gets kind of ugh, stuff's leaking out of it all. Those are microorganisms. They're breaking it down. Those microorganisms are secreting enzymes and breaking it down and returning it to the environment so it can be reutilized again. Those microorganisms are many of them are bacteria, but some of them are in the class of mold, fungi, yeast. They're all basically the same, basically in the fungal family. And they too can be single or multi-cell. Single or multicellular, they too have a cell wall. So they're kind of like a plant. But wait a minute, no chlorophyll. The interesting thing about them is they're what's called saprobes. S-A-P-R-O-B-E-S. And that means they're decomposers. They break down dead stuff. They live off of dead stuff. They secrete enzymes, break it down, and in the process, they return nitrogen to the soil. They return other chemicals to the soil. That's just part of this sort of magical role of microorganisms that we see. Very, very neat. So they're very, very similar. The distinction is the chlorophyll and the algae are water-based. The mold and fungi, those things are principally land-based. And we see them all the time. Okay, I mean that's the kind of stuff. So in the lab, and maybe Dr. Dress will have this, or whoever else is teaching the lab, will have you find something in dorm room refrigerator has been there, moldy stuff, and take a look at that mold under the microscope. Cultivate it. I mean, most of it's in the penicillin family. Okay. Students always have strawberry. That's rhizopus. They have mold have wonderful names. And so, I mean, I had a whole part of my micro class, which is the best class I ever took, was a year-long class. We met 4 days a week. We had lab twice a week, and we had like 3 months of fungi. Because we dealt with it's a foot doctor. We deal with feet and the athlete's foot and all. We got a lot of fungal infections, fungal skin infections, and they have great names. And for somebody like me who likes to show off their memory, it's fun doing that. I gotta have something to show off. I got a vertical leaf of 1/4 of an inch. Your grandma could beat me in a race. I got to practice. So photosynthesis, this is what they do. Chlorophyll, you all know about this. It's a good thing. They take waste products that we put out in the air, water and carbon dioxide. We exhale carbon dioxide. We urinate out water. We exhale water vapors. Plants take this in the presence of sunlight with chlorophyll and transform it to organic compounds, carbohydrates, that you can tinker with and make them into almost anything. And oh, by the way, oxygen. What a country. Go out and thank a plant today. You know? So so microorganisms, that's where the oxygen comes from. Dude, I'm not worried about oxygenic when our planet was evolving, it was different. I mean, it was noxious gases, and that's what they're talking about with the ancient microorganisms are basically the so-called oxygenic photosynthesis. Nothing you have to know about, but again, 70% of the Earth's photosynthesis, which means 70% of our oxygen that we're breathing. So the main forces that drive the structure and content of soil, water, and atmosphere: carbon dioxide, nitric oxide, methane, and all of these gases. And I do not know if that is correct, the NO. They may have meant to put N2. I do not know. So I'm not going to ask you about it because I think that might be a misprint on a PowerPoint. Remember, PowerPoints are not subject to editorial review. And as I look at it now, when they've changed this PowerPoint somewhat, I can see that that's possibly incorrect. But nevertheless, all this, the greenhouse gases and all this stuff, and the climate change and global warming, we won't be having that. We won't be having that for the next week. You remember originally they said global warming. Now it's climate change. I have another term for it. I know I'm being iconoclastic. I'm countering prevailing theory. We call it weather. Bacteria are the most abundant cellular organisms in the oceans. See? Viruses are the most abundant inhabitants of the oceans. That makes no sense. Oh, cellular versus, okay, there are more viruses because viruses are not cellular in nature. So they're just calling them in a generic term. They're things. Bacteria and fungi live in close association with plants in a system obtaining nutrients and water and protect them against disease. That's the beauty from an environmental and ecological standpoint. You can't do something in the planet without impacting it. We build wind turbines as a way of alternatively collecting energy rather than using fossil fuels. The amount of energy that we expend to put them in is extreme. Most of those are built by oil companies anyway, just augmenting the amount of energy they need to produce things. Understand, you can't impact a planet by doing one thing without having something else come from it. I don't teach ecology, but if I was, that's the premise, the whole idea. And so people have to become very, very aware of these things. We all live in close order. If we're going out and eliminating something, then we have problems otherwise. I mean, they said, oh, we're going to deal with this, and we're going to bring in some animal to control a particular population. All of a sudden, we're taking away predators and irregular relationships. For some reason, right now, the Florida Everglades are crawling with pythons, which are not indigenous to there. Somebody brought them in to control something, and now they're taking over. I'm not going. No Everglade vacation for me. And it's cool to drive through the Everglades if you haven't done so. It's a very interesting area. So, found everywhere on the planet, they're everywhere. Stop. I don't know if that's true or not. So this is the whole idea. I don't have a whole list of these things. All of these, bread, alcohol, cheese, typically they are associated with yeasts, naturally fermenting things. If you leave something out that has something that is fermentable, it's in the air. You can make sourdough starter for bread just by leaving flour and water sitting out. So most of the time, and famous organism, Saccharomyces cerevisiae. So Saccharomyces cerevisiae is, again, ingredients. That's a yeast organism. And just like Candida albicans, when we mentioned things about vaginal yeast infections and thrush and things like that, that's another very common yeast. That's baker's yeast or brewer's yeast. Okay. And I mean, it was early on when they began to harvest honey from bees, which were done thousands and thousands of years ago. The first alcoholic beverage was fermented honey, mead. Okay? And then you started mashing up grains and things like that and let them out and they would ferment. Now, I mean, now obviously we add the yeast directly to them. So if you, I mean, you get those little neat little packets of yeast in the store and you have, you're going to make bread or you're going to make pizza dough or whatever you're going to make with it, right? You do that sort of stuff. It's fun because I need to need. Need, N-E-E-D to Knead, K-N-E-A-D. Nobody thinks you're funny. So all of that, bread, alcohol, we would be nowhere without that. I mean, the name cerveza should ring a bell. That's cerveza. That's what beer in Mexico. Little Dos Equis, Modelo, Corona. I know. Yeah. College students do not drink. Stop. My wife would say it's more funny peculiar than funny ah. Yeah. So I looked this up, mining precious metals. I wasn't aware of that. Cleaning up human created contamination. They have modified bacteria and evolved them over time and sometimes scientifically to clean up oil spills and things like that. So there's bioremediation. I'm not sure about the precious metals, but I'll go backwards for a second. So what I did, and for years, they would put, and I just did a quick Google search on this. But I meant to do it. I mean, then all of these things, microbes and natural antibacterial substances were used to manage wounds long before the discovery of antibiotics. And it's true. People used to put maggots because it would gobble up debris and there'd be microbes on the maggots themselves. They're living things. I know it sounds like yuck, but that's the way they used to do. They used leeches to draw things out. They used to put really bad things. They would put like bird dung on wounds because it would stop the bleeding. The trouble was now you're putting infected material into an open wound, and typically you'd end up. I mean, it was they didn't really understand what they were doing, but they had some good ideas. So they've used a lot of things. Vinegar and wine they'd pour over things because the acidity would kill microorganisms. But a lot of times your microbes are basically your friend. There's only about 2,000 species that cause disease. So the idea of being able to put something with microbes that are non-disease causers is somewhat advantageous. And most of the things, when they had honeys or sugars or different materials, you put on, leaves and things like that, certain ones would be less likely. There was a lot of science behind this. And really, what modern science does, it goes into areas that are unexplored and they do soil samples. That's where they look for antibiotics. Okay, because soil bacteria and soil fungi are where antibiotics are produced. And they go out and examine those things, and they're constantly looking, and you're looking at different, and they're talking to the natives to say, well, how do we use this? I mean, that's when it came to malaria, which is not a bacteria, it's a protozoan, which I'll put up there in a second. When it came to malaria, the number one cause of death in sub-Saharan Africa, not AIDS, malaria. And when we went to dig the Panama Canal back in the late 1800s, malaria was everywhere because we were going through a rainforest. Malaria is everywhere. It's caused by mosquitoes. Okay, it's brought by, not caused by, it's carried by mosquitoes. They're the vector. And we'll get into what vectors are. And the natives said, oh, try this. And they had plants that were called quinine alkaloids. And that's where the treatment came from. So understand that medicine is very interesting in that. So we look at these areas that have been relatively unexplored or untouched. And that's part of the problem that's going on today because we encounter new diseases when we go into the rainforest. Okay, and I'll get and there's a whole lot of examples. I mean, that just gives you some ideas about what I was referring to when I start to talk about that. And I have other notes with regard. And I'll make sure I get everything covered as I sort of wind my way around this particular PowerPoint, as it were. Right on my way, father. So Genetic engineering, oh, I love this. Recombinant, so the bioremediation, microbes already present or introduced intentionally, toxic spills, they do this all the time, oil spills, they have microbes that will surround them and gather the oil spills in so they can be sucked up. I mean, environmental disasters are really problematic, and every so often we have a big one. It's there. I mean, if people are not capable, when you have a big ship and it goes aground and it leaks out stuff, it's big problems. Okay. Because they're not like a vehicle that you can hit the brake if you have enough time and stopping distance. They take a while to turn. Trust me, I know a ship captain. So, recombinant DNA and genetic engineering. Manipulate the genetics of microbes, plants, animals, us, potentially, and create new products. Let's just take a quick look. When we get to bacterial anatomy, let's say a bacteria is kind of like this. Very, very tiny. Other one's kind of like this. Bacteria have little appendages. Not all of them. Kind of hollow made out of proteins called a pilus. Not all of them have that. Bacteria have a big circle of DNA. That's why it's different than our DNA. Bacteria also come with little circles of DNA, and we're going to go over all this again, called plasmids. Plasmids are interesting. Not all bacteria have them. They're what we call non-essential but useful. If it was essential, the bacteria would die without them. So it's not essential to have them, but useful, and typically it carries things like resistance to antibiotics. We're going to get into all this when we talk about bacterial genetics. Interestingly, bacteria can pass useful traits like resistance to antibiotics most of the time merely by making contact. They make contact when this pilus contacts another bacteria, and these little plasmids can go from point A to B. Change it. It's like, let's say you have perfect vision. And you could just by touching her and her and her, we could all discard our glasses and contacts, the whole class overnight. So it's about MRSA. MRSA has a genetic marker to resist a chemical called methicillin. That's what identifies it as methicillin resistant. Staphylococcus aureus. If one MRSA gets into a population, a staphylococcal infection, overnight, it can change it in its entirety. They will all become resistant. How's that? In the same generation, called lateral transfer. When you and I want to get a useful trait, we have to pass it on to our pride. We either have to get it from mom and dad and give it to our kids, assuming we're not like my kids adopted. Just give me headaches. I get headaches. Magic is plasmids. So these little circles of DNA, we can make little strands of DNA. We take the DNA and we can splice it and put it in here. We can actually in the lab sit down and take that plasmid, open it up, insert our own little bit of DNA, and then put it back into the cell here, and we have a very reasonable chance to pass it on to another cell. That's what genetic modifications are about. We can do those because of bacteria. Bacteria make enzymes. One of the ways that they work is they can destroy DNA. They can secrete enzymes and chop DNA. We got a fancy name, okay, for these kinds of things, and it's the foundation, everybody, of DNA analysis. DNA is a giant molecule. The reason we can do maternity testing, crime scene identification, do that Ancestry.com, 23andMe, is because of bacteria. We were able to get enzymes out of bacteria in the lab and chop the DNA, not just chop it randomly, chop it as specific. Because DNA has a very simple language. Remember the A's, the T's, the G's, and the C's, right? So it just chops them in specific areas. And that leaves a way, and we can actually paste stuff to them because we know the specific areas. That's what genetic engineering is about. Most of the time, it's taking plasmids in the laboratory, treating them in such a way, and creating a new lab. We have the ability to actually make the DNA. We do it all the time. There's medicines like it all the time. I mean, many of you going to nursing, if you're going to do labor and delivery, maternal child nursing, we induce labor with what? We have a chemical called pitocin. It's oxytocin, which you're going to learn about in endocrine. It's made in the posterior pituitary, that induces labor. It's a very simple, like 9-amino acid chain. Not hard to make in a lab. But we splice it, and now we're making oxytocin, now we have pitocin. The first genetically modified material that was made, genetically modified, not the organism, we modified the organism, was made with insulin back in 1977. When I was in school, insulin had just made the transition from pork-based because there were a lot of allergies and just dietary concerns about a lot of cultures, Judaism, Islam, that don't just. And they were making it from bovines. It was coming from cows. So again, it's insulin that was coming from, and obviously there would be allergies associated with that, but it was coming from bovines. Insulin, even though it was a more complex protein, about 243 amino acids, they were able to make the active form for human beings. It was called human insulin. All the insulin we give to people today is human. Because there's less reaction to it. That was the first thing we made genetically. That hepatitis B immunization is experiencing some degree of controversy about the ages when it's given these days. Short, that's tons of stuff. A lot of medications, a lot of the stuff you're hearing about today, you're inundated, you're surrounded by it in these TV commercials. I'm sick and tired of hearing, I want to throw something in the TV set every time you talk about something with psoriasis. No, I digress. That's genetic engineering, and it comes from recombinant DNA technology. It makes it possible to transfer genetic material from one organism and another. Listen, this is the magic of what it did. We would not have had this without bacteria. It is just, we don't understand how relevant that has become to our society. Our ability to make all of what we call biologicals today. I was on 2 of them. I don't take them anymore. Okay, I was on one called Humira and one called Cosentyx for my arthritis. I had an immunologically based arthritis. I stopped taking it because I didn't like how it made me feel. Maybe it didn't hurt as much, but I'm getting around. A couple Advil and basically staying active. Other than I spent a lot of time recently throwing things at the TV set because of the steel. And use some creative use of inappropriate words. And trying to keep my wife from hurting herself. So all that stuff's part of it. The vast majority of microorganisms are harmless. They're your buddies. You're covered in them. Staphylococcus is one of them, except it's not Staphylococcus aureus, even though you got a lot of that growing. 2,000 microbes cause disease. We will only suffer 1,999. I'm giving you a break. Joke. 10 billion infections occur across the world every year. You're going to see relatively limitedly in our sphere because we have a lot of things going for it. The state of our health care, the pristine nature of our water supply, all these play a pivotal role. Worldwide is a more significant amount of problems than you can imagine. It's a great chart. Okay, I'm going to take issue. I don't buy that. Okay. Understand, I am not an authority on all of this type of stuff. I'm going to try to give you why we see numbers and understand how they came across. You'll get from our being together that there's a lot of stuff that went on in COVID that weren't necessarily all about healthcare. That's my opinion. Okay. Did you get the COVID shot? I did not. And as I told you just the other day, I'm going to be 75 years old. Still here. So let's understand. If that star is inaccurate, the only condition most clearly caused by in the U.S. data is all this. That encompasses a lot of. If they used to have on this chart before it became politically correct, this was about things like pneumonia, respiratory illnesses. I mean, I can't tell you how many statistics I saw during COVID when a 100-year-old man died, and he died of COVID. He tested positive for COVID. He was 100 years old. The fact that someone tested positive for it didn't mean it was the proximate cause of death. A lot of pneumonias. It wasn't the only pneumonia and still isn't. So that drives you a little crazy. But consistently, heart disease, cancers, respiratory-related illnesses like pneumonias, of which COVID is one, injuries, stroke, which is cardiovascular, you're going to learn about. This is where they're throwing the chronic lower respiratory diseases. Chronic are people with emphysema and obstructive pulmonary disease, which you're going to learn about in A&P. Okay, this is This would be the sort of the infectious disease. So I think they've skewed this erroneously. Alzheimer's, okay, diabetes, a lot of complications with that, liver diseases, kidney disease, etc. I'll buy all of that. I mean, I find this to be a little troubling that they've blanketed and think that's the only thing that's ever killed people. Okay, I'm not disputing that there were a lot of people who died during this when it first appeared. That was the worst, and that is very consistent when we look at coronaviruses. As they mutate, they spread more easily, they become less severe. That's a very consistent finding with corona-based viruses. Just to throw the information at you. So understand, you're going to get a lot of my spin, and please, I'm not a researcher in this. I'm just someone who studied it a lot. So this is the last part I'll give you today. There's a lot, and this is the idea between what we call emergent and resurgent diseases or re-emerging diseases. Emergent are mostly from mutations. This was a mutation. Corona, and we'll go into how it mutated. AIDS probably was a combination, and there's a lot of good evidence between a failed attempt to make polio vaccine in sub-Saharan Africa under terrible conditions. I'll give you chapter and verse on this. Hepatitis C is again a variation. This is an RNA virus. RNA viruses mutate. This is where we went into. Zika was discovered when they went into the Amazon for the Olympics and the World Cup in 2014 and 2016 in Brazil. They went into the rainforest, and all of a sudden people are getting sick from something called Zika, which is a mosquito-borne disease in a particular viral category. West Nile, very, very similar. Okay, tuberculosis is really the significant player with re-emergent or resurgent diseases. And I don't want to give it short shift, so I will explain it to you either virtually on Monday or if we meet on Monday, and certainly give you the rest of that information. Thank you. Have a great weekend. Please, safe. We're starting on Saturday afternoon, Saturday evening. It ain't going to be a good idea to be driving around too much, everybody.